Hi guys, welcome back to my YouTube channel, and in today's video we're going to talk about fat acid synthesis. This is going to be a beast of a topic, so please grab a piece of paper and a pen and then take down points. But before we go further, please guys, as always, go down and subscribe. Make sure you like and comment. You don't know how I feel when you do that. I always appreciate it. So let's get started. When we talk about fat acid synthesis, what is coming in our minds? So this is self-explanatory. So fat acid synthesis, we're simply talking about making new fat acids. So we are making new fat acids. And this is very important because fat acids, this is a storage form of acetyl-CoA. Now, when is it taking place? So fat acid synthesis is taking place during warfare state. So it is taking place during warfare state. Now, what is warfare state? So warfare state, this is after eating that meal rich in carbohydrates. So let's say after eating in Shima, that is warfare state. Now, where is it taking place? That's a wonderful question. So it's taking place in the cytoplasm. Cytoplasm of the cell. It is different from beta oxidation. If you remember, beta oxidation, this is the breakdown of fat acids. It takes place in the mitochondria, but fat acid synthesis, which is the making of new fat acids, it takes place in the cytoplasm. Fat acid synthesis, primarily it takes place in three main cells, or three main types of cells. We have got the liver cells, or hepatocytes. We have got skeletal muscle cells, or myocytes. And we have got the fat cells, or adipocytes. So it's taking place in the cytoplasm of the hepatocytes, myocytes, and adipocytes. Now, what is involved in fat acid synthesis? So fat acid synthesis, we are making fat acids using what we call malonyl-CoA. So we are using malonyl-CoA. So malonyl-CoA, this is the building block for fat acid synthesis. And this malonyl-CoA, what you should know is that it is coming from acetyl-CoA. So malonyl-CoA is made from acetyl-CoA. And we know that this acetyl-CoA, it is coming from what we call pyruvate. And the pyruvate, it is coming from glucose. And this glucose, it is coming from the meal that we eat. So during warfare state, what's going to happen? The level of glucose is going to increase. High level of glucose is going to increase the amount of pyruvate, which will also increase the amount of acetyl-CoA in the cell. Acetyl-CoA, when it increases, some of it will enter TCA cycle, as we know it. But in TCA cycle, we're going to produce a lot of ATP. And ATP is an inhibitor of TCA cycle. So when you produce ATP, you're going to inhibit TCA cycle. Now, what direction is the excess acetyl-CoA going to take? So when TCA cycle is inhibited, the excess acetyl-CoA will go in fat acid synthesis. It will be converted to malonyl-CoA, and malonyl-CoA is a building block for fat acid synthesis. So now, let us look at the TCA cycle briefly and understand what's happening. Let's imagine that we have got a mitochondria, and pyruvate is entering in the mitochondria, where it's converted to acetyl-CoA. And this is called oxidative decarboxylation. The conversion of pyruvate to acetyl-CoA is oxidative decarboxylation. Now, acetyl-CoA is going to combine with what we call oxaloacetate, so that we can form a very important compound that we call citrate. This is a TCA cycle that's happening in the mitochondria. The enzyme that catalyzes this reaction, we call it citrate synthase. Now, what will happen to citrate? Citrate is going to be converted to another compound that we call isocitrate. And this reaction is reversible. What do I mean reversible? you can convert isocitrate back to citrate by the same enzyme. This enzyme that is here is called aconitase. So aconitase can convert citrate to isocitrate, and it can also convert isocitrate back to citrate. The third step of TCA cycle, we are converting isocitrate to alpha ketoglutarate. And this is a regulatory step. So in this step, we are converting isocitrate to alpha ketoglutarate. And the enzyme that we have here is called isocitrate dehydrogenase. Isocitrate dehydrogenase. This is a very important enzyme because it is well regulated in TCA cycle. The main regulator of isocitrate dehydrogenase, it is ATP. So when you have got a lot of acetyl-CoA, you are going to produce a lot of ATP in the TCA cycle. And ATP is going to inhibit this enzyme called isocitrate dehydrogenase. And you're going to block that step. Therefore, TCA cycle cannot continue. So in other words, during well-fed state, you end up producing a lot of ATP, and that ATP will eventually inhibit TCA cycle. Because the priority of the body is to make ATP, but when we make ATP, it's going to inhibit TCA cycle. The product inhibits the pathway. That's something that you should remember. Now, what do you think is going to happen when you inhibit TCA cycle at that point? Isocitrate will start building up in the mitochondria. But thank God, this step, it is reversible. So isocitrate, instead of building up, it can be converted back to citrate by the same enzyme called aconitase. And also don't forget that we have this acetyl-CoA which is coming in, because this is during well-fed state. You are producing a lot of acetyl-CoA. TCA cycle is inhibited, but acetyl-CoA will continue flowing in, and it will continue being converted to citrate. So I've got isocitrate being converted to citrate, and acetyl-CoA being converted to citrate. And don't forget that when you're converting acetyl-CoA to citrate, we remove a coenzyme A. So what's going to happen at the end is that we're going to have a high concentration of citrate in the mitochondria. So citrate is going to build up because acetyl-CoA is flowing in and is being converted to citrate, and then isocitrate is also being converted back to citrate. So citrate level will increase. Now, the good thing about citrate is that citrate can cross the mitochondrial membrane. Therefore, it's going to start moving from the mitochondria to the cytoplasm, and it's going to cross via what we call the citrate shuttle. So citrate will be taken to the cytoplasm, and that is why it's going to be used to make fat acids. So something that you should get here, that acetyl-CoA cannot cross the mitochondrial membrane. So this cannot cross mitochondrial membranes. Therefore, acetyl-CoA is going to be converted to citrate. Then citrate is going to go to the cytoplasm because it can cross the mitochondrial membrane. Now, what's happening in the cytoplasm? So imagine that this is the mitochondria and we have citrate in the mitochondria. So citrate is going to be transported to the cytoplasm via the proteins that are here. So we have got translocase proteins that are found in the mitochondrial membrane. They are going to transport that excess citrate that is building up in the mitochondria to cytoplasm. System of proteins that transport citrate from mitochondria to cytoplasm is called citrate shuttle. Now, what will happen to citrate in the cytoplasm? Citrate in the cytoplasm is going to be broken down. Remember what was making citrate? Citrate was being made from acetyl-CoA and oxaloacetate. So even here in the cytoplasm, citrate will be broken down to acetyl-CoA and oxaloacetate. But also don't forget that for us to convert acetyl-CoA and oxaloacetate to citrate, we are removing a coenzyme A. So here we must add that coenzyme A back so that we form acetyl-CoA and oxaloacetate. So citrate will be broken down into two things. Acetyl-CoA, this is the cytoplasm, and oxaloacetate. We are adding a coenzyme A, and we're using ATP. So here in the cytoplasm, we are regenerating acetyl-CoA. Now, why didn't we just get acetyl-CoA from mitochondria and bring it outside here? The answer is simple, because acetyl-CoA cannot cross the mitochondrial membrane. Therefore, it is converted to citrate because citrate can cross via the citrate shuttle, and then in the cytoplasm, citrate will be converted back to acetyl-CoA in oxaloacetate. Now, what's going to happen to oxaloacetate is that it can be converted to mallate. The enzyme that we have here is called mallate dehydrogenase. And we are going to use NADH. Then mallate, it can be converted to pyruvate. And the enzyme that is here, it is very important. So the enzyme that is here, that's converting mallate to pyruvate, it is called Malic enzyme. And at this point, we are going to make NADPH. So Malic enzyme is converting mallate to pyruvate, and we are producing NADPH. This is one of the few steps in the body that produces NADPH. The other one is the pentose phosphate pathway. So malic enzyme is converting mallate to pyruvate, and pyruvate, it can go back in the mitochondria where it's going to be converted to acetyl-CoA, and the process can repeat, or it can go in TCA cycle. Now, having looked at the fate of oxaloacetate, let us look at the fate of acetyl-CoA. What's going to happen to acetyl-CoA? So acetyl-CoA is going to be converted to another compound that we call malonyl-CoA. And this is very, very important. Malonyl-CoA, this is actually the building block for fat acids. When we are making fat acids, which we are going to see, we are going to be using malonyl-CoA for us to make fat acids. Like when you're making a house, you use blocks. Same applies to building fat acids. When you're building fat acids, we're going to use malonyl-CoA as the building blocks. Now, here we have an enzyme that we call acetyl-CoA carboxylase. It is called acetyl-CoA carboxylase because what we have here, it is a carboxylation reaction. And what do we mean carboxylation reaction? We simply mean we are adding a carbon dioxide. The other thing that we are using at this step is that we are using ATP, breaking down ATP and forming ADP. The other thing that is very important about this acetyl-CoA carboxylase enzyme that you should know is that acetyl-CoA carboxylase, it requires a cofactor that we call biotin. It is also known as vitamin B7. So biotin, that is a cofactor. It is also known as vitamin B7. Now, why am I focusing much on acetyl-CoA carboxylase? This step, this is a regulatory step. Please, you can forget other things, but don't forget this step. So acetyl-CoA carboxylase is well regulated, and this is the regulatory step of fat acid synthesis, the conversion of acetyl-CoA to malonyl-CoA by acetyl-CoA carboxylase. Now, what regulates this enzyme? Let us look at what's regulating this enzyme. When you look at this, we can see that we have got this enzyme here that we are calling SCC. This is acetyl-CoA carboxylase. This is an enzyme that converts acetyl-CoA to malonyl-CoA. Now, this enzyme is very important and it's well regulated. Now, what you should know is that some enzymes in the body, they are active when they don't have phosphate groups. Or in other words, they are active when they are dephosphorylated. So as to carboxylase, this enzyme it is active when it has no phosphate groups. But when it has phosphate groups, it means that it is not active. Now, what is going to activate this acetyl-CoA-carboxylase? Let us look at what's going to activate it and what's going to inhibit it. The main activator of acetyl-CoA-carboxylase, it is an hormone that we call insulin. So insulin, it is an anabolic hormone. What do we mean anabolic hormone? It promotes building up of biomolecules. So for example, when you've got insulin, you're going to start building glycogen, you're going to start building fat acids. So insulin promotes building, while glucagon promotes breakdown. That's why insulin is called anabolic hormone. Glucagon is a catabolic hormone. So insulin, it activates the building of fat acids by activating this enzyme. Now remember, this enzyme is activated when it has no phosphate groups, or when it loses phosphate groups, it's going to be activated. So this hormone insulin, how does it act? Or how does it operate? So insulin is going to activate what we call protein phosphatase 1. This is an enzyme. So imagine that ACC, it is inactive. Imagine that it's not active. It's having phosphate groups. Because I said that when it's having phosphate groups, it means it's not active. So when ACC has phosphate groups, it means it's not active. So how is insulin going to activate that enzyme? Insulin first will start by activating PP1, protein phosphatase 1, and then protein phosphatase 1 is going to start chopping off those phosphate groups from acetyl-CoA-carboxylase enzyme. In other words, protein phosphatase 1 is going to dephosphorylate acetyl-CoA-carboxylase. So start removing those phosphate groups. When we remove those phosphate groups, you're going to find that this enzyme is going to be active because it is active when it has no phosphate groups. So PP1 is going to remove those phosphate groups and it will activate acetyl-CoA-carboxylase. So when they ask you, how does insulin activate acetyl-CoA-carboxylase? Insulin, it's going to activate protein phosphatase 1, and protein phosphatase 1 will start dephosphorylating acetyl-CoA-carboxylase and eventually activate it. What else is going to activate this enzyme? We also do have another important compound. This compound, it is called citrate. So citrate is going to come and bind on the allosteric site. So citrate will bind on the allosteric site. And it's going to activate this enzyme. So we call it as an allosteric activator. So an allosteric site is simply another site on an enzyme other than the main active site. So when citrate binds to the allosteric site, it's going to activate the enzyme because citrate, we know that it's the one that is forming acetyl-CoA. And acetyl-CoA will form malonyl-CoA. When we have a lot of citrate, what does it mean? So it is simply telling us that TCA cycle is inhibited because when TCA cycle is inhibited, acetyl-CoA is converted to citrate, as well as a citrate is converted to citrate. So a lot of citrate, it's a signal that TCA cycle is inhibited and that will stimulate fat acid synthesis. How do we stimulate fat acid synthesis? We are going to use that citrate to activate this enzyme called acetyl-CoA-carboxylase. So the main activator of this enzyme, if they ask you, is insulin and citrate. And how do they activate this enzyme? Please just follow what I've explained. Now, what is going to inhibit this enzyme? We have got mainly glucagon. So glucagon, this is a catabolic hormone. And it is produced when you're starving. During prolonged starvation, you're going to be producing a lot of glucagon. So glucagon, it promotes breakdown of compounds or biomolecules. So when you're starving, we don't have acetyl-CoA. So the body will start using fat acids. So it will release glucagon, which will promote the breakdown of fat acids, and it's going to inhibit the build of new fat acids. So glucagon inhibits fat acid synthesis by inhibiting this enzyme called acetyl-CoA carboxylase. Now, how does glucagon inhibit this enzyme? So glucagon, when it goes to the cells, it binds to the receptors that we call G protein coupled receptors. Now, when it binds to these receptors, it's going to activate the CAMP pathway or the CAMP pathway. And then eventually will activate an enzyme that is called protein kinase A. Now, protein kinase A, this is an enzyme that phosphorylates other molecules. So protein kinase A is going to add phosphate groups to this enzyme, which we call acetyl-CoA carboxylase. And when we add phosphate groups, what does it mean? This enzyme means it's going to be inhibited. Because remember I said that this enzyme is active when it has no phosphate groups, and it is inhibited when it has phosphate groups. So glucagon, it inhibits this enzyme by binding to the G protein coupled receptors, and via the CAMP pathway, it's going to activate protein kinase A, which will phosphorylate this enzyme, and this enzyme will become inhibited. What else inhibits this enzyme? We have got hormones like cortisol produced by the adrenal gland, norepinephrine, and epinephrine. We also have another thing that can inhibit this enzyme, and that is called long-chain fat acids. So long-chain fat acids, these are allostatic inhibitor of acetyl-CoA carboxylase. So long-chain fat acids, these are the products of fat acid synthesis. So when you're building fat acids, you're going to make long-chain fat acids. Now, long-chain fat acids can come and inhibit this enzyme when they're in excess. In other words, look what was happening in TCA cycle. When you produce a lot of ATP, ATP itself will inhibit TCA cycle to say, we are enough. It is enough. Stop producing any more ATP. The same applies to this pathway. In fat acid synthesis, when you produce a lot of fat acids themselves, they will inhibit this enzyme acetyl-CoA carboxylase, and that will result in the inhibition of fat acid synthesis. So, in a nutshell, this is what I have on the regulation part. The activator is insulin and citrate. The inhibitor is glucagon, cortisol, epinephrine, and long-chain fat acids. This is very important for multiple choice as well as for essay questions. Please don't forget this part. When we make malonyl-CoA from acetyl-CoA, malonyl-CoA is very important. And for multiple choice question, please don't forget this. Malonyl-CoA, it inhibits an enzyme that we call CPT1. CPT1, this was an enzyme that was found in beta oxidation. Beta oxidation, this is a breakdown of fat acids. So CPT1, this is an enzyme that facilitates the transport of fat acids from the cytoplasm to the mitochondria for beta oxidation or to be broken down. So malonyl-CoA, this is an inhibitor of CPT1, carnitine palmitoyl transferase 1. Why are we inhibiting fat acids when we are making fat acids? The reason is to prevent futile reactions. Futile reactions, these are simply two or in the opposite reactions taking place at the same time. So when you're making fat acids, we don't want to start breaking down fat acids at the same time, because it's like you're building a house and then someone is demolishing it. It means you're wasting energy. So the body is made in such a way that when you're making fat acids, you're going to produce malonyl-CoA, and malonyl-CoA will inhibit the breakdown of fat acids by inhibiting CPT1 enzyme. So for multiple choice question, please don't forget that CPT1 is inhibited by malonyl-CoA. Now, malonyl-CoA is the building block of fat acids. Now, at this point, what have we made? Let's look at what we have so that we can start building fat acids. Because what we've talked about so far, we're simply making building blocks for fat acids. We haven't yet started building fat acids. So let's look at what we have. So at this stage, we do have three things. The number one thing that we have is malonyl CoA. And malonyl CoA, we say this is the building block of fat acids. Second, we have main acetyl CoA, which is also very important in the making of fat acids. We also have NADPH, which is coming from where? From the pentose phosphate pathway and also from the conversion of malate to pyruvate. NADPH is very important in fat acid synthesis. You're gonna see how this happens. So guys, I'm gonna end here in this video. The main purpose of this video was to talk about how we are making malonyl CoA as well as NADPH. So we've talked about that and we have understood that. So in the next video, I'm going to talk about the main fat acid synthesis pathway and what's going to happen. So please go and check out part 2. It's very important. Thank you so much for watching and please don't forget to subscribe.